Paul Verschure and Connie Prescott. And Partha was describing to us an amazing project where you have this sort of massive automated data collection on a very detailed anatomical level that can lay a foundation for all sorts of new insights in the brain. So Partha, why don't you try to give us a short summary how you ended up doing this?
I was really shocked to find out how little we know about the how the brain is wired. That's what got me started. I am trained as a theoretical physicist, and when I came into biology, I'm slowly coming to grips with the knowledge that we are very much limited by lack of information. And we have this paradox that we are data rich and data poor at the same time. We have lots of data, but sometimes critical pieces are missing. Once I realized that even in the most thoroughly studied model organism, which is the rat, the wiring diagram of the brain at a scale that neuroanatomists have studied has large empirical gaps in it. I thought I would set out to try to rectify that situation.
But in some sense, it's very counterintuitive because often in neuroscience, also when you talk with other neuroscientists, it's this feeling like, well, we're drowning in data. We have so much data at different scales, different species, physiology, anatomy, etc. So how do you position that with respect to this data richness? So how do you now deal with this paradox?
I think the way I think about it is that we are data rich. You're quite right. There's more than 500,000 articles being published abstracts being published on PubMed every year. So there's a tremendous amount of information out there. I think this information is very heterogeneous and not really integrated. There is no super brain that really has all the information.
And so was you.
So yes, we are data rich in this very heterogeneous sense. Lots of little bits of data. We are data poor in the sense, in neuroscience, and compared with, let us say, in genomics, where we have comprehensive information about a certain scale of organization. Let's say the genome. We've got the full genome of multiple species. But we can't say the same at any scale, really, of any organism for the nervous system, that we have the full set of information, with the exception of C. elegans, which has been kind of the example that people have put out. So for me, the reconciliation is that on the one hand, we do have a lot of heterogeneous, unintegrated information, sometimes partial, because people have focused, over-specialized on certain areas of the system. A lot of studies of the hippocampus, or a lot of studies of the visual system. So if one takes an integrated perspective and does a project where you map out, as in our case, the whole brain circuit at some level of resolution, yes, that's generating a significant data set. But at the same time, it's not even at the scale of this large heterogeneous data that's out there.
But given how you summarize this argument, you're facing two challenges in some sense, no? Because on the one hand, isn't one implication that you say, well, yes, we have all this data, but we might as well have not collected it because it's not organized in the right way. Is that really the consequence of what you're saying? Would you agree?
Yeah, I mean, I think that's one way of putting it. I think a good analogy that is certainly not new at this point is thinking about Google Earth. We probably have a lot of information about geography, about, restaurants in a particular city and so on and so forth. But until there was a framework on which to hang all these pieces of information, they remain disparate. So perhaps this project that we've initiated, one thing that it will do is to provide a framework in which to collect this heterogeneous information. If that succeeds, then, we will resolve the paradox to some extent.
Underlying your argument, though, is another assumption, which is really interesting, because in some sense you're saying, well, the Human Genome Project was actually a great success. And I want to follow this paradigm now from neuroscience. But is that really a reasonable assumption?
I wouldn't quite, put it in those terms. I would say that yes, it was a success. But success as defined in certain ways, right? I think one way in which it succeeded is providing this integrative framework for pulling information together. Another way in which it succeeded was it helped us gain understanding of the evolution of genomes. We've also gained information about human history, right? So we haven't solved certain problems which were promised, and maybe we will still solve those problems in the future, but in certain ways it has been successful. And I think in those ways is where I would, want to borrow from the success or hope for the success.
So looking ahead to when you've built this new huge data set, what's your idea about how people are going to use it? I mean, I can see sort of neuroanatomists looking at the raw data and say, yeah, this fills the gap. I don't any longer need to do that experiment because I can just look at your database. But for those people that want to draw something out of it, to condense it down into some more compact understanding of the brain, what kind of tools do you think they will be able to use on this database?
Well, we will try to provide integrated versions of the data at lower resolutions. So in the ideal world, what we should be able to provide is a probabilistic map saying that if you start from point A in a reference brain, and you were a neuron with a cell body near point A, what's the probability that you had a projection or, an arbor at point B? So that sort of information integrated across the injections, we will be able to provide. And, that will then presumably help people who are trying to gain an understanding of the architecture of the whole brain circuit. I mean, I know that this is a nascent field in the sense that not a lot of people are trying to do that. There's more interested in understanding the microcircuits in a particular region of the brain, right?
Yeah.
But we do hope to provide some integrated pieces of information. Apart from guiding, thinking about how the system is organized and how it works, my hope is that we will also get multiple species. So we will be able to address evolutionary questions. Previously, brain evolution has been studied largely in the context of shapes and sizes. So, cortex has grown in size, the olfactory bulb has shrunk in size, and so on. But we haven't really studied it at the level of what the circuits are and how they have changed, with exceptions. But it has been very much driven by non-circuit considerations, the evolution. I don't know if you agree with me.
Well, what I was wondering in that respect is also the incredible individual variability you might find in your data. So how are you going to handle that, right? So you might take, let's say, a mouse or a rat or a monkey, from which you get your anatomical data. But now you know that over individuals, there will be an incredible dispersion of these kinds of projections. So how do you see yourself extracting the rules from that kind of variability?
I do have a premise that the variability will not be so large that we cannot extract these regularities. So going back to a comment you made during my presentation is that I do have a hypothesis, in some sense, a weak hypothesis, that there is something to be learned from these experiments because there is some underlying organization that is relatively conserved from individual to individual in this mouse strain. We will do multiple injections and multiple repeats of the same injections in the same brain region. So empirically, we will hope to answer this question. I don't think it's a theoretical question. So I do have a theoretical hypothesis that the variation will be controlled enough that there is a meaningful mesocircuit. But we will try to prove it empirically.
And at how many different, since the brain is continuously changing also ontogenetically, so how many measurement points do you think you would need in a single species?
We are doing it in the mouse at a single age. We have not yet even started thinking about a developmental version of this. One thing that I'm hoping is that, yes, we are running this particular experimental project in my lab, but we are hoping to demonstrate that this kind of project can be done quite economically in multiple laboratories. So once that comes about, I think people should be able to fill in multiple ontogenetic steps. What we are hoping to do is to take one age and then look at different mouse strains. So different genetic constructs and see how the circuit changed at a given age.
Right. So there's something interesting about the trajectory that you went through. So as you said earlier, you started as a theoretical physicist, and then sort of discovering the brain, if you want, and in some sense now becoming much more an experimentalist in some sense.
Yeah.
But then let's say a large scale anatomist or industrial scale anatomist. And if I look at the way you present the argument, it seems to be also interesting that along the way you've been shedding more and more of your theoretical skin, right?
I do it at night privately.
Exactly. Is that fair to say?
No, my bedtime reading recently has been, formal logic and models of real computation. I mean, I haven't shed my theoretical interests. It's just that I have become much more conservative than when I started in making theoretical models, because I've come to realize how difficult the problems really are that we are trying to describe. And what happened to me when I was a theoretical physicist is I, developed a great admiration of theory. And so I don't want to, take the name lightly, so to speak.
It's very interesting. So in some sense, what you're saying is that in this move from, let's say, the naive but maybe very ambitious theoretical person into now, let's say, the anatomist at the bench, you have acquired more humility towards the problem.
Absolutely. Absolutely.
That's the point.
Absolutely. Yeah. I think that these are very hard problems, and I still hope to make some theoretical contributions, but I would like them to be solid contributions.
So where do you think we are in terms of theories of the brain? I mean, what aspects of the brain do we understand well, and where do we need to pay attention to?
I think single neuron biophysics is a clear success story, to the extent that there's a well-developed theoretical framework, and that has got an attached experimental framework, and there's a close feedback loop between the theory and experiment. Some other parts, I think it's piecemeal. I think certain systems in the brain are well developed theoretically. I'm not saying that they are fully developed, but the sensory systems are relatively well developed in that way. We have a good, at least, approach to the visual system or the auditory system. Also to a lesser extent, but also to motor systems. At the peripheral levels, I think we've got good models. What I feel is lacking is more systems level understanding of the whole system, and I think that the theoretical work that has been done there is non-mathematical, but still legitimate theorizing. And people who have done that are perhaps not thought of as theorists because they've really thought about the system. They've articulated their thoughts, right? I would still call that theory. I mean, Darwin didn't have a single equation, right? So, what is the status? I think that there is a lot of work to be done. Progress seems to me to be pragmatically occurring in fields which are tied into robotics. Insect locomotion is one area that I've followed to some extent, and it seems to me to be working quite well. Of course, that's limited in the sense of understanding the brain. But one lesson that I've drawn from there is really to think about engineering theories and to think about what the system does, what's the logic that one would employ in constructing systems like that, and then trying to understand whether the evolutionary processes are compatible with such thinking. Is there, role for convergent evolution? So if I were to say in a single sentence where I see the future of theory, I would say take engineering much more seriously. I think that it's certainly been a theme that has been explored since the times of cybernetics. It has perhaps been sidelined in some areas of neuroscientific theorizing, but I think it needs to be brought more to the fore, and more training is necessary in engineering disciplines of the some of the neuroscientific theorists.
Are you saying that we should give up this more physics-based dream of, let's say, closed form solutions of the phenomena, that we're trying to study and go more for let's say open solutions or descriptions of these systems. Is that the key step there?
Yeah, I think that there has been a certain limitation in the theory community, at least those of us who came from a physics background, to take a point of view that resembled too closely the successes of theoretical physics and simply hoping that the same trick will work again. I think that that's not working is really the evidence. That there are some isolated examples like Hodgkin-Huxley equation, partial differential equation describing how the action potential works, might lead us to believe that in general, ordinary and partial differential equations are what we are looking for in terms of theoretical constructs. Again, looking in a forward modeling perspective, observing some physical phenomena in the brain and then modeling with ordinary or partial differential equations. Seems to me that approach, in spite of being very successful in one domain, has not generalized. And it seems to me one needs a multiplicity of theoretical framework. So I have coined this motto. I say ontological monism and epistemological pluralism. So there is only one reality out there. We all agree.
You always win at Scrabble, I understand.
I don't think there's a disembodied, soul that is driving my brain. But at the same time, I think there are multiple legitimate descriptions, epistemologies, ways of theorizing, modes of theorizing. They may use different mathematical tools. And it is not true that one reduces to the other. I think this is an assumption people have made. So when people talk about reductionism, the discussion is a bit confused because it's not clear whether they're talking about physical phenomena reducing to each other. That doesn't make any sense to me because there's only one phenomenon. The question is whether theories reduce to each other. And sometimes they do, sometimes they don't. And I think one has to be a little more Catholic in.
So you take a very Kuhnian approach here.
I would like to, take a more Catholic approach to theorizing. Yeah. And that is definitely new for me. I mean, I did not come from a tradition where that is done, although having trained as a condensed matter theorist, I was perhaps more open to it than if I were, let's say, trained in particle physics.
Why do you think the mesoscopic scale is such an important scale to understand the brain at, as opposed to other scales? Or is it just that we haven't done enough work on that level? Yeah,
I think that it is a scale at which the brain needs to be understood and a scale in which understanding is comparatively lacking. I'm also inspired to study this scale because it seems to be developmentally patterned. So it is in the genetic code, so to speak. Given that there is a juggernaut out there, which is kind of genome biology, and we have learned, certainly human knowledge has progressed in that domain. It would be great if we could link up neuroscience with that progress. And one scale where I see that link occurring quite distinctly is at this mesoscale, because the mesoscale is developmentally patterned out of the genome, or at least that would be the quote-unquote hypothesis.
So the heterogeneity at the mesoscopic scale, there might be, within elements of that scale, there might be more homogeneity and then potential for using learning algorithms or developmental algorithms to wire up systems.
Right. So the hypothesis would be that, let's say the genome patterns this mesoscale.
Yeah.
With environmental input, but let's say the genome, that's understood in modern day discussions, right? But without making those caveats, let us just say the genome patterns the mesoscale.
Yeah.
Then variations in the mesoscale are related to genomic variations. Then one should be able to relate those two things if one looks across species or if one looks across individuals. There could be also environmental perturbations that cause,
right,
fluctuations at the mesoscale, but, the idea is that it's genomically sculpted. And then variations at a more microscale, certainly some of that patterning is also due to, rules. Let's say one neuron has to connect to another neuron.
So kind of self-organization, would you hope that would give us leverage at the smaller scale?
Right. Yeah. One would expect that self-organization is playing a more important role at the smaller scale. So this would be a way of separating where the developmental program is more important from where, the learning self-organization rules are more important.
But what's the metric of this mesoscopic scale exactly?
I don't think there is a fixed length scale that I would associate, because if you look in different parts of the brain, there are different sizes. But I would simply point to classical neuroanatomical atlases, where, the sizes of regions or nuclei, vary if you look across the brain. They could get down to, depending on what brain you're looking at, in the mouse brain, they could get down to hundreds of microns, but they could also be larger, millimeters. So,
don't you think though that we have to refine or improve this definition? Certainly, if we want to do a comparative anatomy.
Yeah, I think it has to be data driven. I don't think there is a definition I can give today in advance of having gathered the data set. What I hope is, after we have data sets like this, and we already have some existence proof from the gene expression data set from the Allen Institute, that one can maybe empirically extract these correlation lengths or scales directly by looking at the data. What we can hypothesize based on neuroanatomical literature is that such scales exist. And good guesses as to what those scales are in specific brain regions.
Now, one thing you said that earlier is that you felt that, let's say, the genome would be controlling development at just one of these scales, while, let's say, these lower levels would be more self-organizing. Why are you saying that?
Well, it's a guess. I cannot articulate all my, previous knowledge that leads to the guess. But one reason is you look at different mice, you get the same atlas. This is just a statement about how the community is organized in this research. If it was the case that there was tremendous environmental influence on the scale at which classical neuroanatomical atlases are organized, we would probably not be able to use atlases, right? So the very fact that we can use these atlases, to me, is some indication that these are not specifically environmentally sculpted.
But this could also be a bias of how we treat these phenomena. No? Like in psychology, it's a typical problem that, okay, we moved into statistics because we cannot understand humans at an individual level. On the other hand, now in psychology, people are bumping into this limitation of statistical approach because actually there is not this normative person. So maybe for your atlases, you're facing a similar kind of problem, no?
I would certainly agree that there is not a normative mouse brain, the Platonic brain. But what I do believe that there's a low dimensional manifold of brains, that it's not a as high dimensional a space as, let's say, the number of neurons in the brain would lead us to believe. So even though there is variability, and that variability could well be developmental plasticity that has environmental impact on it in the same way that we would expect in psychology. But I at least have a strong hypothesis that this pace of variation is quite constrained. It's quite small. And I would also say it differs from region to region in the brain, and we will probably find that out. This is an empirical question. We will be doing these studies in multiple mice. So we will be able to look across, individuals and really ask the question empirically. I think that's the way to ask it.
What's your feeling then about this low-dimensional manifold? And certainly if you look also across species, or even including species that do not exist anymore, I mean, what's this common template there? What's your feeling about this? So do you see, are you expecting to converge to, say, a common design template for all vertebrates, for instance? Or
it's a very intriguing thought, right? That there are, whether due to common ancestry or due to convergent evolution, in some sense, selecting for the same circuit because the same function has to be subserved. It's a very intriguing hypothesis, which I hope is right. That there are these common design rules or templates that we will find. It may not turn out to be true, but that would be the interesting hypothesis in my mind. And it's an empirical hypothesis. I would say that I hope it is true because there is certainly some theoretical biases I have that are pushing me in that direction. I think the system wouldn't work otherwise.
Well, given that we have a finite set of genes building the brain,
there you go. I mean, there is certainly some developmental rules, and I think that we have other examples like our musculoskeletal system that are also patterned, about which we don't ask these questions, right? So is the brain really that different from all the other organs in the body at the level, let's say, of this mesocircuit?
So your idea is to do a lot of the work in your own laboratory at Cold Harbor, but you're trying to get other people on board this project because by the sounds of it, it expands to include many other species, also to include animals at different ages. There's a huge work program here. Why do you think people should prioritize this as opposed to all the other things that we might be doing in the neuroscience?
Well, I guess it's a free war. People have to choose for themselves. No. But more seriously, I think that, I don't know, talking about humility, I've just come to realize that the problems are extremely hard. So I would like to work on areas where clearly progress can be made. Now that can be very dangerous. One can get into the instrumental fallacy, right? But, I have tried to articulate some of the arguments in the position paper that we put together that I would draw people's attention to in PLOS Computational Biology that came out last year with the first author, Jay Boland, and the last author being myself. But just to, try to lay out some motivations, one great motivation is that we are at the technological cusp where this has just become doable within the funding constraints of individual laboratory. I think that's for me a great motivation. That was not true a few years ago, simply due to memory costs. Today it is doable.
Yeah, but this is the because we can argument. So
because we can argument. I think it's an important argument because there are
many elections with that,
Yeah.
That's it.
No, I mean, it's I think it's an important argument. But the, second argument, I think, is that, the scope of discovery, I think, is large. We simply don't have these circuits, at a very simple level. How are we going to understand how the brain works? The brain is a circuit. We don't have the circuit. People could argue we know certain bits and pieces of it. But that's a very simple argument, One can try to refine the argument, but that's one argument. And then there's a third argument, which is this information integration that we were talking about. That we are battling this huge deluge of data and information, how to tie it together. Well, here's a way we could. And it's clearly a project that is much bigger than something that I could handle in my laboratory. So I'm definitely hoping that other people will want to join in. We are very open, by the way, with our data set, even before publication, when the data will officially be made public. If people would like to collaborate with us at the level of data analysis, we're happy to do that. If people would like to learn how to do these experiments, or if they're interested in setting up their own pipelines, we're also very happy to actually provide that input and expertise. Because I think that the realities is that we'll publish a 2-3 page paper, let us say, but that won't summarize what's going on. No. Things are changing in that way.
However, one thing I was wondering about now is whether you're not stepping a bit too easily over this challenge that also Tony put in front of you. That imagine, so now we have this whole federation of laboratories contributing to this pipeline, and they all have their own species to work on or whatever. You will have a huge problem integrating all this data, because also at some point people would like to bring in other kinds of data, physiological data, behavioral data. Do you already have a taxonomy in place to bring all that information together in an integrated form? Or you think that's just going to self-organize?
It's work in progress. Some people are working on it. I do think it has to be to some extent data driven. And talking about theory, here is one place I think theorists really need to pitch in. Because taxonomy is really a theory about the system. Once you start dividing what the parts are and naming them, you've kind of made your mind up about how to divide the system and how to name them.
But isn't it a scary thought to actually now open the floodgates, but you don't have the buckets ready yet to catch the water?
I think that there are some starting efforts. So it's not as bad as it could be in other fields, because in anatomy there has been a scholarly tradition, and so there is certainly a good head start, I would say. But you're right. I mean, it is a challenge. I think that we will be helped simply by having unified data sets. That this taxonomy problem, what we want to call a region, will be less important than having a coordinate where you can really say that, well, I call it A, you call it B, but we are all referring to the same objective artifact. And that's what will help that taxonomy problem.
So what was amazing in the Human Genome Project is that there was this expectation that it would take quite some time to sequence human genome. And then actually some people really industrialized this whole process. And it turned out it could be done just in a few years, which was astonishing, even though it's still debatable how informative all this data is. Now, how many man years is it going to take you to get the data for, let's say, a single mouse species? How many man years are we talking about?
Well, we are hoping to release a draft next fall. And there's about, now what I mean by...
I meant complete, right? Everything.
Everything will take infinitely long by definition.
Really? The whole brain.
But I think on the scale of 2 to 3 years, we will have, given existing projects, we will have a quote unquote mesocircuit. Even next year we will, in every laboratory order 10 people maybe. But
so you have 30 man years. You said 30 man years to get one mesocircuit of one species done.
I think that's reasonable.
Okay.
But we have defined mesocircuit in a particular way. People are going to challenge that and have arguments about it. I think it's a doable project on a short time scale.
And for yourself, I mean, is there going to come a point where you say, okay, I've got enough data of a sufficient quality that I now go back and be a theorist about this data?
It's happening already.
Oh, okay.
In the sense that even when trying to interpret our very first data sets or in trying to analyze them, the problems that come up are definitely of a conceptual theoretical nature. I'll give you an example. So this question came up about quantifying connection strength. And you often see isolated neurons in, different parts of the brain showing up. So if you have hundreds or thousands or, tens of thousands of neurons in a particular, let's say, region of the brain that you have injected, and if one neuron shows up in some other part of the brain, in some sense, the circuit doesn't have this clean organization that we theoretically started out with, that it has got these like tails. How do you even think theoretically about a circuit like that? What are those individual neurons doing? Are they artifacts of the fact that biology is not a clean engineer? Or was it a reason? Is it a feature or a bug?
Yeah.
But now in this endeavor, in which also your project got supported to go after this connectome of the brain, there are also other initiatives underway.
Yes.
Where people would look more at, let's say, what they would call them the effective connectivity of the brain, based on data sets that are of a very different nature, using different kinds of imaging techniques and so on. So do you expect a convergence among these different approaches, or do you think that's just too far in the future?
I have a theoretical disagreement with, and I'll be very frank about this, about the term functional connectivity being used to describe what are temporal correlations. So simply because two time series are correlated, and this we have known for a long time, does not mean that there is a, let's say anatomical connection between those two points. So I don't see any reason why there should be a quote unquote convergence. it's certainly true that the anatomical connectivity is going to drive correlations, but I don't see the reverse route.
So the correlations are a necessary condition of the causal structure, right? But it's not the
of the anatomical structure. And I strongly advocate calling correlations. Yeah, I think it is misleading and perhaps even distracting to call it connectivity.
Okay. Your database will be monosynaptic, will it?
Our database is really of neuronal morphology. Don't really have synapses.
Oh, okay. But you'll be just looking along a single
one neuron
fiber. And so it'll still be a problem for me if I want to know if there's a connection between A and B that might go via C. You know, I can tell there's a projection that goes into C, a projection from C into B, but I can't know from your database that those two things meet up. Yeah.
Correct. And there are other limitations to this project, namely we are not being cell type specific.
Yeah.
There are other projects which are coming about which will try to be more cell type specific and yet get, neuroanatomical connectivity in place. I regard this particular mesocircuit that we are mapping out as operationally defined, because it's defined in terms of retrograde and anterograde injections and whole brain imaging object. But there will be missing pieces of information that will then have to be layered onto this thing. More projects.
But now, also in your presentation, also in your work, you have been spending quite some time to look at gene expression patterns in the brain.
Yeah.
And do you see that as giving you leverage in understanding these anatomical pathways that you're trying to reveal right now, or do you see this really as separate databases as well?
No, I see the connection that they are both indicating a mesostructure, so to speak. They are getting us objectively at this intermediate length scale that is there in neuroanatomy. The gene expression data sets are probably giving us indirectly information about cell types. I don't think they are directly telling us about connections. But yes, they are definitely connected.
So within this gene expression data you showed, there was something very puzzling because anatomically, at least if I listen to the anatomist, I'm not an anatomist myself, they would always tell you, look, cortex, relatively uniform, well-structured, modular, etc. And if you go to subcortical areas, things get more messy. It's more variable, heterogeneous, and so on. Right? Mm-hmm. And if I look at your gene expression data, so you made the point. Actually, you have less genes expressed at the subcortical levels than at the cortical level. So what do you make of this apparent contradiction?
I actually think it's the other way around. I think the subcortical structures are more homogeneously structured and more carefully sculpted than the...
No, you will have much more interspersion of cell populations, also the kind of transmitter systems.
Correct. So they are more... Yeah, I see what you're saying. They are... It's more messy in the sense that the modules are not well separated, but it may be less variable. So, yes, it is it will be messy for us to disentangle, but it will be less variable, I think, from animal to animal.
So sometimes what you're saying is maybe cortex is so neatly organized because there are just more genes being expressed there to keep that clean structure in place, while you would have then less genes expressed subcortically, leading to more of a dispersion of how these cells actually anchor themselves in the substrate and so on. Is that how I should interpret what you're saying?
That's an interesting hypothesis. I guess cortex is not translationally homogeneous, as we know from Brodmann areas, and the gene expression patterns certainly show that there are different cortical structures.
Cool. So look, I have two questions to finish up. So, if we had to define the Partha law of science, right, of our attempts to understand the brain, what would be this Partha Mitra law of investigation and understanding? What's the law we should adhere to?
Law. Well, I made my aphorism, right? There's one reality with multiple theoretical approaches. I think we have to be open-minded about disciplines, and this sounds like a truism, but and it sounds really something that people say as a slogan. But I really think that we need to educate ourselves in different disciplines, like engineering disciplines or neuroscientists need to learn more about population biology and evolution and so on and so forth.
Perfect. And then, so if I go visit you again in Cold Spring Harbor 5 years from now, I'm going to say, okay, there's this one prediction you gave to me today, and today I'm going to check whether it's true or not. What's this one prediction you're willing to stick your neck out today?
Well, that's a very tough one.
Come on, you're a tough guy.
I guess this notion that the mesocircuit is genetically wired and that genetic polymorphisms will cause alterations in the mesocircuitry that may be correlated with neuropsychiatric disorders, which are also, have genetic predispositions. That's for me, especially the affective systems that are related to emotional behaviors. That for me is sort of the, I don't know if it's a prediction, but that's a story that I hope in 5 years to really understand and gain insight into our behaviors.
Wonderful. Partha Mitra, thank you very much.
Thank you.